Okay, so today we're going to talk about different topics related to cancer. So generally, when we talk about cancer, we talk about cell proliferation that is not under control, but it's not only that. We are going to, when the tumors grow, we are going to cover this class and in the following, the genetic components and how we have been studying that. And if you see, there is something very interesting is if you see here, there are vessels, the process of angiogenesis, that you form these new blood vessels because the tumor grows and it needs to eat. So one way to prevent cancer from spreading is to go here and prevent this process of angiogenesis. If the tumor doesn't have oxygen or doesn't have nutrients, is going to die. And we're going to talk about also about movement. Yeah, another way to prevent cancer is to try to prevent cells from moving or accumulating mutation. And in this case, here this cell, if you see, is going inside of the blood vessel and is going to travel, and from there is going to take a highway into another body parts that are far away from the primary tumor. And we're going to cover all these topics. Okay, so let's start. And in the beginning, I'm going to still give you an introduction to cancer. And the first thing that I want to tell you is that why you have one thing that you need to learn today is why we study cancer. Yeah, why this is so important. And I'm going to show you statistics about it. Then I'm going to tell you, okay, but what actually is cancer that we started introducing in the previous class. Then we're going to divide it in three sections. One, that is chromosomal rearrangement, that is more from a genetic perspective. Then transformation, what happened when a cell modified and transformed into a cancer cell. And then we're going to move into different stages of cancer. Okay, so these three main ideas. And finally, we're going to talk about how the cancers move, yeah, and later we're going to talk about some of the the genetic basis of cancer, and this will take us several lectures, and there is a lot of more information that we can cover. But today we're going to start talking about what is an oncogene. Okay, and finally, there will be another video lecture with an expert from the University of California, San Francisco, that you can watch, and he will give you a very detailed um, lecture about the history in cancer that I enjoyed a lot. So if you have any doubt, let me know, but let's start. Okay, so let's start with this first question. Why we study cancer? And again, I remind you, this is the video lecture that you should watch after this class. Really good one. There is three sections. We're going to start with this one. And he's an expert on cancer, and he has developed different treatments for cancer, and he's an expert on the history of cancer. Okay, so I'm going to show you statistics and in multiple classes. Some of them are newer than the other ones, but the idea is pretty similar. For example, if you compare to the States, any organization, the Canadian American Society or the American one have very similar numbers. For example, this one is something that grabbed my attention, that every minute there will be 3 new cases in the States and 1 new death because of cancer. So is very interesting to think in this term. And in Canada, things are very similar. Yeah. So in Canada, in our lifetime, 2 out of 5 Canadians will develop cancer, and 1 out of 4 will die of cancer. Okay. So during our lifetime, the 40% of the Canadians will develop cancer and 25% will die of cancer. And why? Because Canada is such a peaceful country. There is a good healthcare. there was vaccination. Most of the people have clean water, so people is not dying because of a war. Is dying because of aging. Yeah, aging related disease, and one of those is cancer. Yeah, so it's positive and negative to think about that. Yeah, so kind of so peaceful that people is dying just because they are getting very old, and one of those diseases is cancer. So, and you will hear this multiple times. I said it last time. Soon, cancer will become the leading cause of death. And globally, there is every year there is 8 million, more than 8 million people dying from cancer. And and what we were talking about, that if is cancer increasing, remember that I said, like, we always think that, oh my God, there is so much pollution that the cancer should be increasing all over the planet. We're eating so many pesticides in the food. However, what is happening is that actually cancer is slightly reducing, but it's not reducing as much as, for example, heart disease, yeah, cardiovascular disease. So, for example, this is the age-adjusted mortality rate between 1975 and 2005, and from there was a 70% reduction from death with mortality with cardiovascular diseases. However, for cancer, we still need to do more work. It's 10 times, almost 10 times lower. Yeah, and this thing continue being like that. Yeah, that still cancer is the second leading cause of death between diseases. Still, cardiovascular disease is still the highest one, but there has been a huge improvement. Yeah, and the question that we always have is like, why? And we talked last time about that. Is because cancer is not one disease; it's multiple diseases, hundreds of diseases altogether that show the same symptoms, that is abnormal cell growth, because of multiple reasons. So So as I said, much higher reduction in mortality rate in cardiovascular diseases than in cancer, and is something that is still happening right now. Uh, so if you see overall cancer death rates from the 1999 to 2015, there was a decrease of 1.4 per year in women and a decrease of 1.8 per year in men. Yeah, so still, there is a lot of work to do, but things are not as negative as they were in the past. What is surprising is that we have improved a lot the quality of life for other diseases. However, for cancer, there is more work to do. And in the video lecture, he will tell you a little bit more of the statistics of the states. It's still the same. And every time that you talk about cancer, you start always with this. You will see many professors working on cancer in this video lecture, and they will tell you relatively similar statistics. Okay, so I already finished with this section, that is why we need to study cancer, and now you know that 2 out of 5 Canadians, yeah, is expected to develop cancer in their lifetime, and 1 out of 4 will die for cancer. Again, this is when they get very old. Okay, so age is very important, but still is something that we need to work. So now I'm going to again go on and talk a little bit what is cancer. And every time that we talk about what is cancer, we're going to give more and more details. First in the genetics in the first courses, and then we will talk about the cell biology. Okay, so to study what is cancer, I love to show this cell culture. I have shown it to you in other classes. These are HeLa cell that we are going to talk today, and there will be parts on the quizzes talking about this. So please give it a look and give it a look. Each one is a cell. Each one, the bright section are cells that are going to divide, is the membrane, yeah. And inside you see the nucleus, yeah. So similar to the previous class, we were talking about number of cells, the presence of the cytoplasm, the regularity of the cells, and the sizes of the nucleus. So pay attention to, and you see what happened here. So look, that cell is dividing, that one also is dividing. So look about this. Here you have a cell with two nucleus already. Yeah, so that is something very weird that, well, all the other cells have one nucleus, this one has two nucleus. Yeah, and if you see, you start finding that there are cells, these cells is giant compared to other ones. Yeah, so and I wonder, think what happened when these cells is going to divide. Yeah, if how many nucleus will have the daughter cells? And look this one again. How big is this one? What do you think that happened when this one divide? Yeah. So here, and what do you think that is happening with the DNA? You know, we are just seeing the division. Do you see what happened? There was a division of two. Instead of having two cells, there was three cells. Yeah. So let's, yeah. So go very fast. This big cell, yeah. Instead of it, you see that instead of going into divided into two, it went into three, while other ones went into divided into two, and you start having more and more weird things. Look this one. Look at this one. Look what happened. There's one cell with two nucleus, and when it divides, it went into three cells and very weird nucleus. Yes, this. So the idea is that you start making a lot of mistakes when, or not, the cells start making a lot of mistakes, and the nucleus start becoming very regular. The cytoplasm, the sizes of the cells. There is so much the things that have changed. So if you remember, I'm connecting this topic with the previous class. Okay, so question. I wonder you think about this. Are the cell normal or not? And I wonder you think why. So write for the assignment, take notes and you can hand it at the end of the class. Okay, what are the things that you saw that they were abnormal? Okay, so what do you think about the number of cells and the sizes of the cells? What do you see about not only, oh sorry, here it was the nuclei. How many nuclei they should have? How many, the sizes of the nuclei? Now regarding to the cells, how many cells do you see? Do you see any difference in the sizes? And the cell division, we generally talk about that there is the cells are identical during mitosis, yeah. However, I show you that here even the cell cycle is disrupted. Okay, so just write all those things and remember these are HeLa cells. And during the class and what we have done in many research projects is trying to analyze if the cell dynamics can be a useful tool to differentiate normal from cancerous epithelial. I know some of you have studied the cell death, other ones have studied the frequency of oscillations of the cells, and all of that is to relate to this, to why the cells start behaving differently. Okay, so now we know that, and just to remind you, cancer is not one disease, yeah? Instead of that, cancer is a given name to more than 100 diseases, yeah, that are related. And what they are related, so if you see here, you have the most common type of cancer that we said that it was the carcinoma. So here you have a carcinoma is developing. Here you have an epithelium, and in blue you start seeing a tumor. Yeah, and it's going into a transition from a normal to a normal epithelium to a carcinoma. And why all these cancer have in common? Yeah, so you can have here For example, cervical cancer, it will be lymphoma, stomach cancer, breast cancer, all of these ones. Even between them, you can have different genes affecting to create prostate cancer. Yeah, there are different reasons why prostate cancer can appear, and and sometimes even one prostate cancer from a person is very different from the other one, and it will be more similar to a cervical cancer, especially if, for example, from the yeah, it moved from one region to another one. Yeah, so if this was the primary tumor, this will be more similar. Yeah, there could be more connections here from primary to secondary tumors. Yeah, rather than the location of the cancer. So, and there is a lot of variation that we will talk during class. Okay, so what I just told you, that in all type of cancers, some body cells begin to divide without stopping and spread into surrounding tissues. They could, and we will talk about very different genetic pathways involved in the presence of different type of cancer. However, all of them have these two traits. They divide without stopping and they spread to other tissues. And in class with this professor, he will tell you an idea, this similar idea, but in more detail, that when in cardiovascular diseases, you have a single cause and a single disease. In cancer, you have multiple causes and multiple diseases. So it's something that we now have very clear, and we have been mapping the genes and the dynamics of the cells and the tissues, how they change. And this is something that historically it seems that it is a fact and is not going to change. That cancer will be, there will be multiple diseases grouped into one single name. Okay, and now let's focus on a little bit of the cell biology. So we're going to be jumping between genetics, cell biology, development, and on different fields. And one of them is in cell biology, that cancer can be understood as that cancer cell can be understood as a misregulation of the cell cycle. And I know that you have talked about the cell cycle since maybe high school, elementary school. You have seen mitosis and the cell cycle in diversity of life, in genetics, and different courses. I'm not going to talk a lot about that, but a misregulation of a cell cycle is one of the key components. Later during the semester, we will see a specific proteins involved into that. So you have the DNA, you have a normal cell, and remember, in the DNA, you have, in the nucleus, you have DNA, and you have different genes. Yeah, and what happened here is that if the cell divisions, you start forming, instead of identical cells, you start forming these irregular three dividing cells at the same time. You're not going to have the same number of genes. So what is going to happen is that, while in normal cells you have a uniform nucleus, this is going to ensure that normal cells have the same genes and the same amount of DNA. Yeah. However, if you during cancer, this is something that is lost. And remember, during the cell cycle, you have the instruction to make an exact copy of each one of those cells. And this is lost when you start losing all those genes, and when you not only lose the genes, but also you change the cellular environment. Yeah. So those two things will be important, and we will cover in the second section of the course how the cellular environment is, and in particular we call that microenvironment. Yeah, the environment surrounding the cell is very important for the production of cancer. Okay, so I love to show you videos. Here is a video that I found that I think it summarizes everything that I have
and starts
said
to multiply out of control.
up to now.
The DNA is copied, but there is a mistake. The cell checks whether everything is okay with the DNA. Is it damaged? Has it been copied correctly? Here you can see it's damaged. Usually, a cell with damaged DNA will die, but sometimes the cell ignores this warning and continues. The cell gets ready to divide and builds a scaffold called a spindle, which attaches to the DNA. The cell checks again that everything is okay. Is the DNA attached properly? Here you can see that part of the damaged DNA is attached to the wrong spindle. Usually, a cell with a faulty spindle will die, but sometimes the cell ignores the warning and divides anyway. The cell splits, but each new cell doesn't have the right number of chromosomes. So some cells don't have the correct number of genes, and others have extra copies. These faulty cells no longer have the correct instructions, so they start to multiply out of control.
Okay, so in case that you need to find this video, it's from Cancer Research from the UK. So this is generally when we talk about cancer, they always show this schematic that there you have normal cells that they will divide and they have the same DNA and they know when to stop. Yeah, and here the opposite happen, and as they show in the video, there is no control. Okay, so we are going to talk about now we know that what is cancer, we know why it's important, and we talk about that DNA has changed, and we talk about cell division that is in check. So now later in class, at the end of the class, we will talk about what is an oncogene, yeah? That we're going, and in the following class, we will talk about tumor suppressors. So some specific genes that not all the gene is going to produce a cancer, yeah? Change. It could just produce cell death, it could produce different things, but there are specific genes that are in charge of controlling these two aspects, how the cells grow and how the cells divide. And when there is mutation in those genes is when cancer appears, and we have identified hundreds of those genes. Another process that it will be very important and that we will cover later in class is the topic of apoptosis. And Esri and Emma and Tyson and Ella, they start a project on this, trying to understand apoptosis. And it's just fascinating. Why? Because apoptosis is just fundamental. It's not only that the cells divide, it's not only that there is growth factor, it's also that in any healthy tissue, the cells need to eventually die. Yeah, when they don't die, they can start accumulating mistakes. Yeah, so in a cancer cell, if you remember the video that I show you, no matter if the cells were very big, if they didn't have all the correct amount of DNA, if they have two nucleus, they will just continue dividing. Yeah, so this is something that that is common in cancer. That you have ways to control the damage in cell, and the cells will die. Yeah, and in cancer cells, this control is lost. Okay, so uh, just pay a lot of attention. This will be in your quiz. In this class, the professor is going to give you very good historical uh, view, yeah, or summary of the history of cancer. And he, for example, he will talk about this, about Rudolf Virchow, and I think we talk about this with some of you in generic revolution. On if you remember, cell biology is a very recent field, and and something that they found is that metastasis, yeah. So when tumors spread, that is the process of metastasis. Those new tumors, as I said before, they resemble the primary tumor. So it's like a process of evolution, you know, that you have there is one tumor and it moves into a different region, but the tumor number two, it will look the new tumors, it will move and they will have a common ancestor and they will move to a third place. Yeah, and maybe this one will die, but if you see, you can create and I have shown you multiple times that you can create phylogenetic trees using the DNA of different tumors. Yeah, so and if you remember, this still preserve this law that all cells come from a different cell, and that it was proposed here in the when we start thinking about cell biology. Okay, and the professor will give you a more detailed analysis of that. Okay, now I want to give you some tools that they will be important for you to understand the following video lectures, and some of you already have heard about that. I just want to remind you. So let's start with this question of the quiz that maybe you already took, and remember you can take it multiple times. So the question is, which of the following refer to studies conducted outside of a living organism, typically in a laboratory setting? So here, studies that are conducted outside of a living organism, and that are generally happening in, here is very important, in a lab. Yeah, so these two things are laboratory and conducted outside. So those are the key things. So they could be, so I want you to think which one of these will be. What is in situ, what is in silico, what is in vivo, what is in vitro. Remember that all of these concepts should be in italics, yeah, when you write them. And they don't need to have capital letter. They should be like this, but they should be idolized like in italics. And here, because it's PowerPoint, it made it capital, and in Moodle it will not appear in italics, but it should be in italics. Okay, so let's remember, some of you already know the answer. There are at least three experimental approaches, although they could be combinations, and they could be more, but at least I want that you remember these three. In silico is a type of approach, experimental approach, that is performed in a virtual setting. Yeah, so for example, all the work that Candice does, somebody before work with the proteins, measure all the forces, measure no, more than measure, sequence the different amino acids, and they give all the information, and then she modeled the proteins, how they fold. Yeah, so this is an in silico that is mainly in a virtual setting. Are computer simulations, yeah, are all those things. In vitro, yeah, as you can see here, that it means in glass is when you take something that is not alive, that some tissue that is not in the organism, so to put it outside to work in a lab. And here you can have a petri dish, yeah, you can have also in a test tube. You have so many examples of things here. You can have a cell culture in the petri dish, like the HeLa cells that I showed you in the beginning, that you study different lines that they can grow there. And this is one that we will discuss today, is in vitro, in the HeLa cells that you already observed. Later we will talk about that in a second. And finally, the one that you have done with me, that is the in vivo, that is when you study an organism that is alive, that a tumor grows right away in the organism. You don't take the tumor outside and put it in a petri dish, or you study the development of a leg, but you don't remove the leg and put it in a petri dish, or you do simulations. You mainly study the organism while it's alive. Okay, so three approaches that we talk about: experimental, in silico, computer simulation; in vitro, that in petri dishes, that is when you take pieces of tumor or a piece of a tissue outside of the organism; and in vivo is when you do it with organisms that are alive. Okay, so here, pretty easy, is in vitro. Why? Because these are conducted outside of the living organism, so it's not in vivo, definitely. It's not a computer simulation. And later you can start Googling what is in situ. in situ that we will talk about that. And this is typically in a lab setting. Okay, so there will be multiple questions about this topic. Remember that we talk about HeLa cell is because those cells came from a person. Yeah, this is why they are called HeLa cell, and this professor is going to talk about that in more detail. But they are very important cells that have been used in so many experiments. Now I'm going to just show you again a sample video that make life easier, and so later you understand when he talk in a more scientific way. I'm just going to introduce that with a video.
Imagine something small enough to float on a particle of dust that holds the keys to understanding cancer, virology, and genetics. Luckily for us, such a thing exists in the form of trillions upon trillions of human lab-grown cells called HeLa. Let's take a step back for a second. Scientists grow human cells in the lab to study how they function, understand how diseases develop, and test new treatments without endangering patients. To make sure that they can repeat these experiments over and over and compare the results with other scientists, they need huge populations of identical cells that can duplicate themselves faithfully for years. But until 1951, all human cell lines that researchers tried to grow had died after a few days. Then a Johns Hopkins scientist named George Guy received a sample of a strange-looking tumor. Dark purple, shiny, jelly-like. This sample was special. Some of its cells just kept dividing and dividing and dividing. When individual cells died, generations of copies took their place and thrived. The result was an endless source of identical cells that's still around today, the very first immortal human cell line. Guy labeled it HeLa after the patient with the unusual tumor, Henrietta Lacks. Born on a tobacco farm in Virginia, she lived in Baltimore with her husband and 5 children. She died of aggressive cervical cancer a few months after her tumor's cells were harvested, and she never knew about them. So what's so special about the cells from Henrietta Lacks that lets them survive when other cell lines die? The short answer is, we don't entirely know. Normal human cells have built-in control mechanisms. They can divide about 50 times before they self-destruct in a process called apoptosis. This prevents the propagation of genetic errors that creep in after repeated rounds of division. But cancer cells ignore these signals, dividing indefinitely and crowding out normal cells. Still, most cell lines eventually die off, especially outside the human body. Not HeLa, though, and that's the part we can't yet explain. Regardless, when Dr. Guy realized he had the first immortal line of human cells, he sent samples to labs all over the world. Soon the world's first cell production facility was churning out 6 trillion HeLa cells a week. And scientists put them to work, in an ethically problematic way, building careers and fortunes off of Henrietta's cells without her or her family's consent, or even knowledge until decades later. The polio epidemic was at its peak in the early 50s. HeLa cells, which easily took up and replicated the virus, allowed Jonas Salk to test his vaccine. They've been used to study diseases including measles, mumps, HIV, and Ebola. We know that human cells have 46 chromosomes because a scientist working with HeLa discovered a chemical that makes chromosomes visible. HeLa cells themselves actually have around 80 highly mutated chromosomes. HeLa cells were the first to be cloned. They've traveled to outer space. Telomerase, an enzyme that helps cancer cells evade destruction by repairing their DNA, was discovered first in HeLa cells. In an interesting turn of fate, thanks to HeLa, we know that cervical cancer can be caused by a virus called HPV, and now there's a vaccine. HeLa-fueled discoveries have filled thousands of scientific papers, and that number is probably even higher than anyone knows. HeLa cells are so resilient that they can travel on almost any surface. A lab worker's hand, a piece of dust, invading cultures of other cells and taking over like weeds. Countless cures, patents, and discoveries, all made thanks to Henrietta Lacks.
Okay, so this is a very interesting video that I think summarize everything very well and show them now concerning implications of patient care that right now is changing a lot. That you will not be able to take a tumor from somebody else without their consent, and also um, this right now, like for as you see, there is millions and millions of dollars made from these HeLa cells, and the family never made even a penny, and so it's still very controversial.